everyone. Today we are discussing about Chapter 2 of Part 1, Nucleic acid convey genetic information. The idea that DNA might be the key genetic molecule emerged most unexpectedly from studies on pneumonia-causing bacteria, Streptococcus pneumoniae. In 1928, Frederick Griffith experienced that non-virulent strain of bacteria become virulent when they mixed with their heat-killed pathogenic counterparts. The pathogenic strain that is the smooth because of the presence of capsule encoded by the Cap-S allele, and non-pathogenic bacteria become rough because of the absence of capsule and is controlled by the Cap-R allele. On heat killing of pathogenic strain, the genetic components remain undamaged and pass through the cell wall of non-virulent, and after genetic recombination, the non-virulent becomes virulent. The process is called as transformation. Here, in this picture, they have shown that experiment. Here, this is a pathogenic strain of Streptococcus pneumoniae because it is having a capsule around the its body, and inside there we can see the chromosome that is provided with cap S allele, and that cap S allele is responsible for the production of capsule around their body, so they appear smooth and they are pathogenic. And there is another strain of this particular Streptococcus pneumoniae, that is rough strain or rough cell. That means they are devoid of capsule around their body. And that absence of capsule is due to the presence of allele cap R. So there are two strains. Smooth strain that is pathogenic due to the presence of capsule, which is ordered by the allele cap S, and the rough strain, which is non-pathogenic, which is devoid of any capacity of pathogenesis. And this capsular covering, they are devoid of capsule, and that is due to the presence of particular allele cap R. And then this smooth cells or the pathogenic strains of Streptococcus pneumoniae that heat kill, what happens is that their capsular covering will be damaged. But the genetic material, even though it gets fragmented, will not get destroyed. So when this particular heat-killed strains of pathogenic Streptococcus pneumoniae is allowed to grow along with, is mixed with a culture of non-pathogenic strains, that is rough cells, what happens is that the undamaged genetic material that carries this particular allele cap S will be incorporated, or that will pass through the membrane of this rough cell and enters into the cytoplasm. After that, it will process the recombination and cell division to form the smooth cells again. Smooth cells or the pathogenic strains of Streptococcus pneumoniae again. So, even several years after Griffith's observation, biochemists still believe that the transformed agents or the genes were proteins. They were in a confusion that whether this transforming elements are protein or DNA. Okay. So, on 1944, Oswald T. Avery, Colin M. MacLeod, and Maclean McCarthy announced that the genetic principle was DNA and was proved by that experiment using deoxyribonucleics. Okay, so for their experiment, they used different types of enzymes like deoxyribonucleases. We know that the deoxyribonucleases are the enzymes which degrade DNA, and ribonucleases are the enzymes which degrade RNA, and proteolytic enzymes. These are the enzymes that cleaves protein They used three types of enzymes: deoxyribonucleases that can degrade DNA, and ribonucleases that can degrade RNA, and proteolytic enzymes that will degrade proteins. None of these experiments that the deoxyribonucleases can influence the transforming agent. So, when this experiment was conducted by using deoxyribonucleases, they have seen that the capacity of this transformation gets damaged, or the transformation does not happen. But that was not the result during experiment with ribonucleases and proteolytic enzymes. The concept of DNA as genetic material was confirmed by the isotopic study of multiplication of bacteriophage T2 by Alfred D. Hershey and Martha Chase in 1952. Okay, so still the concept was having some confusion, so that confusion kept rectified by experiment using. So the concept of DNA as genetic material was confirmed by the isotopic study of multiplication of T2 bacteriophage by Alfred D. Hershey and Martha Chase. That is Hershey and Chase experiment in 1952. They labeled the protein code of T2 using radioactive isotope, that is S35, and the nucleic acid by using radioactive isotope P38, and followed the fate of phage protein and nucleic acid as phage multiplication procedure. They could notice that much of this parental nucleotide and none of this parental proteins was detected in the progeny phase. So, parental proteins stayed attached to the outside of the bacterial cell, performing no function after the DNA components have passed inside. So, in this picture, this experiment can be explained easily. Okay, so with this picture or diagram, we can understand the concept of that experiment clearly. Okay, so what they did was, first of all, they will label the, the outer coat or the protein coat of this T2 bacteriophage were labeled using the radioactive isotopic sulfur, that is S35, and so the coat protein was labeled using S35, and the inner nucleic acid has been labeled using the radioactive isotope of phosphorus, that is P38. So, and they were mixed with labeled differentially labeled virus T2 phage in a culture of host, and they were allowed to infect that particular host. After that, they are carried out violent agitation. So what happens is that the protein coat that stayed outside the body of this host gets separated, and the P32 label, and we know that the protein ghost, that is the protein coat that remain after the release of nucleic acid cell into the cell of the host. This P32 label nucleic acid only reaches inside the cell. Okay, so that is what they have seen, that they could notice that much of the parental nucleic acid and none of the parental protein inside the progeny phase. So after allowing the multiplication, the progenies get released. When this progeny is released, they could notice that some of these bacteriophages, progeny bacteriophages, where the P32 labeled parental genome, but some others do not possess the P32 labeled duplicate. Okay. So, but none of this possess S35 labeled protein code. that experiment, Hershey and Chase proved that nucleic acid is the material that are responsible for this transformation. Or simply, the nucleic acid is the transforming agent. And some other experiments also prove this concept. Even purified DNA, example, mouse mouse polyomavirus. The nucleic acid of mouse polyomavirus can enter mouse cell and initiate a cycle of viral multiplication and produce thousands of new polyoma particles. Even this purified form of DNA of this mouse polyomavirus is able to infect a mouse cell and initiate a cycle of viral multiplication and pathogens. Okay. While work was proceeding on the X-ray analysis of protein structure, some scientists tried to solve the X-ray diffraction pattern of DNA. At the same time, there was work that has been carried out to, to analyze the X-ray analysis of protein structures. At the same time, some scientists tried to solve the X-ray diffraction pattern of DNA. The first diffraction patterns were taken in 1930 by William Asbury. But the high-quality X-ray diffraction photograph was taken by Morris Wilkins and Rosalind Franklin in 1950. And those photographs suggested the helical structure of DNA, and that was composed of more than one polynucleotide chain, that may be two or three. Okay, so even though different scientists tried to take the X-ray diffraction pattern of DNA, the high-quality X-ray diffraction photograph was taken by Morris Wilkins and Rosalind Franklin in 1950, and from that high-quality X-ray diffraction pattern, that was clear that the helical structure of DNA, and also it is composed of more than two or three polynucleotide chains. And in 1952, a group of organic chemists showed that 3' 5' phosphodiester bonds link together the nucleotides of DNA. So in 1952, a group of organic chemists showed that 3' 5' phosphodiester bond is responsible for the linking of nucleotides of DNA. A complementary double helix was found by Francis H. Crick and James P. Watson in 1953. So, the complementary double helix of DNA was proposed by Francis H. Crick and James P. Watson in 1953. In double helix, the two DNA helix are held together by hydrogen bonds between pairs of bases on the opposite strand. So the hydrogen bonds are responsible for keeping this two strands of double-stranded DNA helix. And the base pairing is very specific. Purine A always pair with pyrimidine T, and purine G always pair with pyrimidine C. In double helix, number of A is equal to number of T. That means number of adenine is equal to number of thymine. And number of guanine is equal to number of cytosine. And that was called as Chargaff rule. Okay, so according to Chargaff rule, number of adenine will be equal to number of thymine in a double helix, and the number of guanine will be equal to number of and vice versa. So the sequence of bases of two chains of a given double helix have a complementary relationship. So the sequence of bases of two strands of a given double helix have a complementary relationship. Then arose the question of how these genes replicate and function. And And the answer was one serves as the specific surface or the template upon which the other strand is made. In this picture, we can see that this is the mode of replication shown by this DNA. Here, the old strand, one of this old strand acts as the template over which this new strand will be synthesized. And then the polymerase that makes the DNA was demonstrated by Arthur Comber by DNA synthesis in cell-free extract of bacteria. Okay, so the involvement of polymerases was made by or demonstrated by Arthur Comber in a cell-free extract of bacteria. Experiments of Comber revealed that the involvement of specific polymerase enzymes, that is DNA polymerase 1 and the energy-rich building precursors or energy-rich precursors like DATP, DGTP, DCTP, DTTP and the nucleotide building blocks of DNA. From this experiment of Kornberg, the involvement of specific DNA polymerase, that is DNA polymerase 1 and the involvement of energy-rich precursors like DATP, DGTP, DCTP, DTTP, and the nucleotide building blocks of DNA. And the DNA polymerase 1 links the nucleotide precursors by 3' 5' phosphodiesterone bond and that works in the presence of a DNA. Okay. The phosphodiesterone. Phosphodiesterone linkage. Here. So the DNA polymerase 1 links the nucleotide precursors by 3' 5' phosphodiesterone bond and that works in the presence of DNA. The permanent separation of two strands during DNA replication was experimentally proved by Matthew Michelson and Franklin W. Starr. So, Michelson Starr experiment using heavy isotope N15 as a tag to differentially label the parental and daughter DNA strands. So, the E. coli. So, during the experiment of Michelson Starr experiment, E. coli were first grown in a medium with N15. Here, the bacteria were. This is a picture that shows the experiment of Michelson Starr. Here, first of all, the bacteria, a group of E. coli, were allowed to grow in a medium of N15. So, after finishing the first generation, they were transferred to a medium with N14. Okay. Instead of N15, they were provided with N14 in the medium. And they were allowed to continue their growth in N14. So they were allowed, after transferring into the medium containing N14, they were allowed to grow in that particular medium itself. And the DNA isolated from bacteria during each stage. DNA was isolated from cell or E. coli from each growth medium. That is, from those in the from bacteria which are grown in the N15 medium, and from those that are transferred to N14 medium, N14 containing medium, transferred to N14 containing medium, and those continually growing N14 medium. Okay. And later on, the DNA mixed with cesium chloride and are centrifuged in an ultracentrifuge. And location of DNA within the centrifuge determined by ultraviolet optics and found that the bacteria grown in N15 had one type of band, that is bacteria that are transferred to N14 medium after one generation in N15, they had one type of band that is a hybrid. That means one band with N15 and other band with N14. Okay, so the ultracentrifugation, this is the DNA that gets separated during this ultracentrifugation. In the case of cells that are transferred to N14 after completing one generation in N15. So, and two generations are okay. And the DNA of this bacteria that were grown in that were continued to grow in N14 were having two types of fat. They were having two types of fat. One is N14 N14 light chain and N15 N14 hydrogen. Okay. The Basis and Stars experiment showed that DNA replication is semi-conservative, in which the single strand of the double helix remain intact during replication process, and during that process that will distribute one parallel strand to each one of the two daughter molecules. Okay. So from this Nicholson stack experiment, proved that or the DNA replication also is of semi-conservative type, in which the single strand of this double helix of this parent will be equally distributed to the offspring. Here, this is a semi-conservative explanation of this. The picture that shows a semi-conservative mode of replication. Here, the red colored one is the parent strand, and during next generation or formation of semi-conservative mode of replication, what happens is that one of these two strands of DNA will be equally given to the progeny, and at the same time, the progeny is able to synthesize a new one based on this uh, strand that has been received from their parent. That's why they are having hybrid DNA. This experiment also ruled out two other models, that is uh, conservative and dispersive scheme of replication. In the conservative model, both of its parental strands remain together and two new strands of DNA formed. Two new strands of DNA formed after an entirely new DNA. Sorry. In the conservative model, both of these parental strands remain together. Here, in this picture, they have shown that these are the parental strands, and both of these parental strands will remain together when during replication, and the new strands will be synthesized, and their new strands will be given to the other. So here, the conservation. This is conservative. Here, it is semi-conservative. Half will be conserved. And dispersive model, DNA were proposed to be broken, and frequently DNA will be uh, DNA was proposed to be broken. This DNA proposed to be broken. This will be broken as frequently every 10 base pair. So after an interval of 10 base pair, this DNA will be broken and used to prime the synthesis of similarly short regions of DNA. And based on each short segment of DNA, a new strand will be synthesized. And these short DNA fragments will subsequently be joined to form complete strands. Okay, so after the synthesis of all the small fragments, based on this all the strands or the fragments of all the strands, after that the entire DNA fragments will be joined to form complete strand. That is why each progeny is getting different segments or the different fragments of both parents and the new one. So in this model, the DNA strand will be composed of both old and new strands. The essence of that much about this Nicholson star experiment. Then the essence of genetic specificity of DNA resides in the linear sequence. Okay, so the essence of genetic specificity of DNA resides in the linear sequence of the four nucleotide building blocks. We know that they are A, G, C, that is adenine, guanine, cytosine, and thymine. And with four letters, the number of potential DNA sequences will be 4 raised to n. N means number of letters in the sequence. For example, bacterial gene, which is made of approximately 1000 base pairs, the number of potential genes of this size will be 4 raised to 1000. So we can calculate number of potential gene sequences by using this equation 4 raised to n, where n is the number of experiments shown that protein synthesis occurs in sites where DNA is absent that ruled out the direct role of DNA for protein synthesis. So, based on some experiments, it was shown that protein synthesis occurs at sites where DNA is absent that ruled out the direct role of DNA for protein synthesis. Okay, so because the protein synthesis does not occur at a region where the DNA is present, that ruled out to the direct role of DNA for protein synthesis. The protein synthesis in all eukaryotic cells occur in the cytoplasm. We know that the protein DNA is located inside the nucleus and the protein synthesis occurs in the cytoplasm. Therefore, at least in eukaryotic cells, second information containing molecule had to exist that obtain its genetic specificity from the DNA. So there should be an intermediate molecule or a second information containing molecule that exists to obtain the specific information from this DNA. This molecule then moves to the cytoplasm to function as a template for protein synthesis, that is the RNA. Okay, so this molecule has to be moved into the cytoplasm to function as a template for protein. It's nothing but the RNA. And the RNA is chemically similar to that of DNA with two differences. Okay. Chemically, the DNA and RNA are similar, but two differences. Only two differences. One is the sugar in DNA is deoxyribose. Here, this is the sugar. The sugar, the base, the phosphate. Okay. This is the picture of deoxythymidine. Here you can see at the second carbon position of sugar. Second carbon position of sugar, there is only H. Okay. That's what is called. There is no oxygen. There is no OH, but instead there is having only a H. That's what is called as deoxythymidine biphosphate or deoxyriboso. But in the case of RNA, the second carbon is having OH. It is called as ribose sugar. Here they have shown uridine. And other difference is that RNA containing no thymine, but instead it contains closely related thymidine uracil. So instead of thymine in DNA, the RNAs are having closely related pyrimidine, that is uracil. Thus, the chromosomal DNA functions as the template for the RNA molecule. Okay, so the DNA which is located in the chromosome is functioning as a template for RNA molecule, which subsequently move to the cytoplasm, where they determine the arrangement of amino acids within the protein. Okay, so the chromosomal DNA functions as a template for RNA molecule. The RNA molecule once formed will move out of this nucleus in the cytoplasm, and there inside the cytoplasm they will determine the arrangement of amino acids within the protein. And in 1956, Franz Crick referred to this pathway or the flow of genetic information as the computer. So in 1956, Francis Crick explained it as the called it as the process of central dogma. Here, the DNA undergoes transcription to form enters into the cytoplasm. The cytoplasm, they will determine the sequence of amino acids of the protein by the process called as translation. Though that later on, this Francis also proposed another concept, that is the adaptive hypothesis. So, through the adaptive hypothesis, Crick proposed that prior to incorporation into the protein, amino acids are first attached to a specific adaptive molecule, which in turn possess unique surfaces that can bind specifically to bases on the RNA template. So, according to the adaptive hypothesis of Francis Crick, it was said that prior to incorporation of protein, the amino acids are first attached to a specific adaptive molecule which possesses unique surfaces that can bind specifically to the bases on RNA template. And scientific community tried to find out this adaptive method. Then comes the discovery of transfer RNA that leads to the discovery of transfer RNA. Okay, so the experiment, in order to identify this adaptive molecule proposed by the Francis Crick, lead to the discovery of transfer RNA. In 1953, Paul C. Samsnick and his collaborators synthesized proteins from radioactively tagged amino acids in cell-free extract and were fractioned by ultracentrifuge, found out that prior to incorporation into the protein, amino acids are first attached to transfer RNA molecules. The tRNA accounts for more than or approximately 10% of cellular RNA. So, according to the radioactively labeled experiments, it was seen that it was nothing but the adaptive molecules are RNA, and they gave the name tRNA or transfer RNA for that particular molecule. Okay, this is a picture of tRNA, adaptive molecule. Okay, which carries this amino acid, and they are having specific sequences that can specifically attach with the sequence on the mRNA. The DNA, after transcription, the mRNA will be synthesized. mRNA will reach the cytoplasm. On reaching the cytoplasm, the adaptive molecule, that is nothing but a tRNA, that is provided with or attached with a particular amino acid, having specific sequence on its other side that can specifically bind with specific sequence on the mRNA. It was previously speculated that his proposed adapter might be a short RNA chain because their bases would be able to base pair and read the appropriate group on the RNA molecule that served as a template for protein synthesis. Francis Crick already suggested that it might be a RNA molecule. The adapter will be an RNA molecule because it is able to specifically bind with the sequences of mRNA. Exact tRNA contains, coming to the details of tRNA, each tRNA contains a sequence of adjacent bases. They are called as anticodons. So it is having sequence of bases, they are called as anticodons, that bind specifically during protein synthesis to successive groups of bases called as codons on RNA template. So the tRNA is having special type of codons or sequences called as anticodons that can specifically bind with codons sequences of mRNA called as codons. Non-specific, okay. And coming to the details of ribosome. Okay, and earlier it was pinpointed the cellular site of protein synthesis as ribosome. So the scientific community tried to explain the non-specific appearance of ribosome. First of all the ribosomes are composed of two unequally sized subunits. So it consists of two unequally sized. One is large and the other one is small. That stick together or fall apart reversible manner depending on the surrounding ionic concentration. So based on the ionic concentration in the surrounding, the two unequal subunits of ribosomes may or may not get separated temporarily. And the second, okay, that was the first concept regarding the ribosome. Second was all RNA chain. All rRNA chain within small subunits are 1500 bases length in E. coli, and in large subunits it was 3000 bases long. Okay, so the ribosome itself is consist of rRNA and proteins, riboprotein. Here, the rRNA, the smaller subunit in the case of E. coli consists of 1500 bases. At the same time, sequences the rRNA chain in the largest subunit of ribosome consists of 3000 bases. And third observation was that the base composition of both the small and large RNA contain the base composition of both the small and large rRNA chain is approximately same, having high GMP content. Then comes the discovery of mRNA. The E. coli cells infected with T4 provided the ideal information that the T4 DNA transcribed to T4 RNA that attached to existing ribosome, and this T4 RNA moves across the surface to bring its base into position where they can bind to the appropriate tRNA amino acid precursor or protein. So the studies on E. coli cells that are infected with T4, they have seen that the T4 DNA first of all transcribed into T4 RNA, and that gets attached to this existing ribosome, and the T4 RNA moves across the surface to bring its base to a position where they can bind with appropriate tRNA amino acid precursor for protein. Nothing but the T4 DNA that gets transcribed to form T4 RNA. The T4 RNA then bind with the ribosome, and they will move its position, or in order to bring its codon exposed to bind with the anticodon of tRNA that is charged with amino acid. Okay. So in so acting, the T4 RNA orders the amino acid and is thus long sought for RNA template for protein. Okay. So in short, the sequences or the codons in mRNA will decide the sequence of amino acids in the protein. Because it carries the information from DNA to ribosome site of protein synthesis, it is called as messenger RNA. So this particular RNA was called mRNA or messenger RNA because it carries the specific information which are present in the DNA into the or that will order the sequence of amino acids in the protein. That is why it is called as messenger RNA. In short, the rRNA component of ribosome, together with some 50 different ribosomal proteins, that is nothing but altogether it will be the ribosome. So in short, rRNA components of ribosome along with ribosomal protein serve as the factories for protein synthesis. So these are the factories over which the protein synthesis is happening, functioning to bring the tRNA amino acid precursors into position where they can read off the information provided by the messenger RNA template. So in short, the rRNA component of this ribosome along with its protein, ribosomal protein, will serve as the factories for protein synthesis, functioning to bring tRNA amino acid precursors into positions where they can read off the information provided by this mRNA template. only a few percentage of the total cellular RNA in mRNA, but they are having varying length and nucleotide composition to encode different proteins found in the cell. Okay, even though only few percent of total cellular RNA in mRNA, but they are having varying length and nucleotide composition to encode different proteins found in the cell. A small segment of mRNA is attached at a given moment to a ribosome. So, a small portion of this mRNA is bind with the ribosome at a time. So, a small portion is attached to the ribosome. A single mRNA molecule can simultaneously be read or translated by several ribosomes, approximately more than 50. That is called as polyribosome. This is the structure of a polyribosome. Here, a single, a small portion of this mRNA is found with a particular ribosome. Here, here, here also you can see the a small portion of mRNA attached to this ribosome, and a series of ribosomes can be bind on this single mRNA. That is called as polyribosome. This is the nascent polypeptide chain that has been synthesized based on the codons which are present over this mRNA. Okay. Then coming to the enzyme. The enzyme that synthesizes or transcribes RNA upon DNA template was called DNA polymerase and was independently isolated by Gerard Hartwitz and Samuel B. P. So the enzyme which is responsible for RNA synthesis or the transcription is RNA polymerase. And the RNA polymerase always works upon a DNA template. And that particular enzyme, RNA polymerase, was first isolated by Gerard Hartwig and Samuel B. P. During transcription, only one of these two strands of DNA is used as a template to make RNA. Okay. Here, out of these two strands, only a single strand of RNA, DNA is used as a template to make RNA. The synthesis of RNA always takes place in 5' 3' direction. And the radio labeling experiment showed that mRNA is synthesized in the nucleus, and within an hour, most of this RNA left the nucleus and are observed in the cytoplasm. So then comes the concept of genetic establishment of specific group of 3 bases, that is follows, determine specific amino acids was learned by biochemical analysis. And the completion of the code of 19 code in 1966 revealed that the genetic code, nothing but the genetic code revealed that 61 out of 64 possible permutated groups corresponded to amino acids with more with most amino acids being encoded by more than one nucleotide. Then arises the triplet concept of genetic codon based on the experiments of Marshall Nivenberg and Erich Mattei in 1961. Okay. They carried out the experiment by adding synthetic polynucleotide poly-U, okay, in a cell-free system capable of making polypeptide chain containing only amino acid phenylalanine. And that experiment conducted by the Nirenberg and Mattei was a breakthrough for the identification of genetic code. So the completion of this genetic code in 1966 revealed that, and the complete set of genetic code has been proposed or completed during 1966, and at that time it was revealed that 61 out of 64 possible permutated groups corresponds to amino acids, with most amino acids being encoded by more than one triplet. Okay, here you can see that phenylalanine is encoded by more than one triplet, that is U and U. Also leucine is encoded by UUA and UUA. Here here the leucine is coded, sorry, here also the leucine is coded by 4 more triplets, and isoleucine by 3, methionine by 1, and valine 4, serine 4, valine 5, proline 4, threonine 4, etc. And pulse labeling could establishes the direction of protein synthesis from amino terminus to carboxy terminus of the gluing peptide chain. So, in order to find out the this uh, amino- direction of this protein synthesis, they used the technique called as pulse labeling. The pulse labeling is in such a way that a few minutes after the initiation of protein synthesis, the cell-free system treated with a radioactive amino acid for a few seconds, and after which the protein synthesis was immediately stopped. So, during pulse labeling, just after the initiation of protein synthesis, the cell-free system was treated with radio amino acids for a few seconds, and after which the protein synthesis was immediately stopped. The polypeptide chain was then treated with trypsin, and the peptide fragments were analyzed for the amount of radioactivity. Since all the proteins finished their synthesis in the presence of radioactive precursors, the peptides last to be synthesized will have the highest density of radiolabeled precursors. Okay, so the final stage of this protein synthesis happened during the presence of radioactive amino acids. So we can expect that the peptides last to be synthesized will have the highest density of radioactive precursors. Conversely, the peptides with least amount of radioactive amino acids would be derived from the region of protein that were first to be synthesized. So, region of peptide that were synthesized at the initial time of this protein synthesis may not have, or that may contain least amount of radioactive amino acids. Investigators observed that the radio labeling was lowest for peptides from amino terminus region of protein and the greatest for peptides from the carboxy terminal region. By combining these two concepts, this led to the conclusion that direction of protein synthesis is from amino terminus to carboxy terminus. Okay, in the beginning itself, it was said that we can expect that since all proteins finished their synthesis in the presence of radioactive precursors, the peptides last to be synthesized will have highest density of radio precursors, and conversely, the peptides with least amount of radioactive isotopes will be derived from the region of protein that were first to be synthesized. Okay, so they have seen that the region, the investigators found that the radio labeling was lowest for peptides from amino terminus region of the protein and greatest in the carboxy terminal region. This led to the conclusion that the direction of protein synthesis is from amino terminus to carboxy terminus. The signals to start or initiate and stop or terminate the translation are present within the DNA itself. Okay, so there is no external signal coming from here, coming from external side, but the signal for starting and stopping this translation resides in the DNA. And three separate columns, that is UAA, UAG, and UGA, known as nonsense columns, because they do not code for or they do not direct the addition of amino acids, and they serve as the translational stop signal. Stop column. At the same time, AUG is the start codon of translation and is coded for methionine residues that are found at the internal protein position. In prokaryotes, the AUG codon that starts new polypeptide chain are preceded by specific purine-rich blobs of nucleotides that serves to attach mRNA to ribosome. Okay, so in prokaryotes, the AUG codon that start new peptide, new polypeptide chains are preceded by specific purine-rich blocks of nucleotides, and that serves to attach mRNA to ribosome. But in eukaryotes, the first AUG always selected as a start site of translation. Okay. So in the case of eukaryotes, the first AUG will be always treated as the start site of translation. Then the era of gene. With the elucidation of central dogma, it becomes clear that how genetic blueprint contained in the nucleotide sequence would determine phenotype. Okay, so by elucidating central dogma, it becomes clear that how the genetic blueprint contained in the nucleotide sequence would determine phenotype. So the DNA sequence can give profound insight into the nature of living things and their evolution. Advent of automated DNA sequencing methods has led to determination of complete genome sequence of hundreds of organisms. So, because of this automated DNA sequencing method, the complete genomes of hundreds of organisms be sequenced. Human genome with more than 3 billion base pairs code for more than 20,000 genes. And the comparative analysis of genome sequence of different groups helps in various ways. By first one, they help to identify important regions of a protein. They also offer insight into DNA sequences that do not encode proteins, and that help in the identification of sequences that direct gene expression, DNA replication, chromosome segregation, recombination, etc. And comparative genomics between different individuals of the same organism helps to identify mutation that leads to disease. And it will be possible to envision a day when comparative genome analysis will reveal basic, basic insights into the origin of complex behavior in humans, such as acquisition of language, mechanism underlying this evolutionary diversification of animal body plans, etc. So we can expect such a um, such a world then this comparative genome analysis will reveal the basic insight into the origin of complex behaviors in humans. Like that, chapter 2 is over. We will meet again with another session. Thank you.